Good day, this is an oral presentation for the course BS934 Gene Technology and Synthetic Biology. My name is Aboyade-Cole and I'm here to present my PCR product, which is TSPO protein for the design and validation of an expression vector. TSPO is an 18-kDa outer mitochondrial membrane protein, which is responsible for the sterol biosynthesis in eukaryotic cells mainly, and as well it regulates the heart rate and contractile force by its interaction with its voltage dependent calcium channels in cardiac myocytes. My aim is to express in Jurkat cells, which are mammalian cells responsible for T-cell leukemia, which is a very rare cancer found in humans mainly, but it can also be expressed in mouse. pET15B is a bacterial expression vector normally found in E. coli. It has a cloning site, 3 cloning sites, and a histag sequence. It also has a restriction site called R1 and an ampicillin resistance, meaning that it can grow when there's presence of ampicillin in the dish in laboratories. There are many findings which state that pET15B can be used for TSPO. The strategy used for it is the recombinant DNA, which is when the TSPO is isolated and as well cut to the right fit, and the fragment of the TSPO is joined by sticky ends, and the insertion of the TSPO is into the host cell, which is the E. coli, and that is when it is transformed into the dish where it is grown in presence of ampicillin. A plasmid whereby it possesses a foreign DNA fragment or a gene which has been inserted into them and is not common amongst a species is known as a recombinant plasmid or recombinant DNA. In this scenario, the recombinant plasmid is inserted into a cell, normally E. coli for this instance, since there are findings which prove that E. coli can be used. This is known as bacterial transformation. Bacteria are employed for both retaining and applying plasmids. Hence, bacterial transformation is crucial for many studies in laboratories to know the genes and to also combat different things like diseases. After the bacterial transformation of the E. coli or the recombinant plasmids found in the Escherichia coli, which possesses the TSPO gene, validation of the vector is the next step, and the most used analysis is the Western blotting analysis as well as the agrogel electrophoresis, which work hand in hand. But the main one is the Western blotting analysis. It is straightforward and it explores the presence of the relative abundance, relative mass, and the existence of the post-translational modifications, and also studies the protein-protein interactions in the plasmids. I have provided findings at the bottom left of the slide 4, which further backs up my statement, which uses E. coli. After the validation of the vector, the recombinant plasmid is then inserted into a mammalian cell, for this instance Jurkat cells from human leukemia cells. This is done by liposuction, which is under transfection. The insertion of foreign DNA into a mammalian cell, mainly a bacterial foreign DNA into a mammalian cell. Liposuction, also known as liposome transfection, is a technique that results in transfer of genetic material into a cell by means of liposomes. The reason is to check if the human leukemia cells is resulted in the TSPO, either accumulation or degradation. And that further concludes my presentation. Thank you for listening and good day.